Okay, well, it's a little bit after 1, so we can go ahead and get started here. And I'm going to be admitting people here. Here we go. So, but anyway, exam 4, obviously we're going to be doing exam 4 tomorrow. The exam is going to be over chapters 12 through 14, so it's not cumulative, like maybe some of your other finals I'm sure are. And it'll take on the exact same exam format that we have had with the previous 3 exams. So just to keep that in mind. In terms of the Q&A that we're running today, I've started a recording, so that's good. And as I've run the last several Q&As, just go ahead and start populating questions here, and I'll get through as many of these questions as I can in this Poll Everywhere document. And of course you can vote on the ones that you want to be addressed right away. And of course we already have some there. So if you have your own questions, go ahead and put them there. I'll come back periodically to this, and I will try and get the chat open here. Let me see if I can't get the chat. But if people can just tell me that, oh, there's a comment in the chat, of course you can unmute yourself and say, hey, there's a comment in the chat, or if you have a question, and you don't mind speaking aloud to all of us, go ahead and do that as well. I welcome those things. So, okay, so we'll probably run this. We'll see where we are, about 1:50 to 2, and then I'll let you on your merry way, and we'll try and get through as much as we can here. So, all right, so let's go here. So first, look at this. It says practice exam short answers. I'm not surprising that would be a question. So I'm going to come here and be able to write on all this. And I have the practice exam pulled up. So let's go ahead and look at this practice exam short answer, as it looks like there's quite a bit of you that would like to go over this. And it says this: in his studies of alcoholic fermentation by yeast, Louis Pasteur noted that the sudden addition of oxygen to a previously anaerobic culture of fermenting grape juice resulted in a dramatic decrease in the rate of glucose consumption. This effect has been called the Pasteur effect and can be counteracted by the addition of 2,4-dinitrophenol DMP, an uncoupler of oxidative phosphorylation. And so the first question is this: why would yeast cells consume less glucose in the presence of oxygen? So let's go ahead and diagram this, because I think that this really speaks to a lot of what we're doing in the class. And so there we go. And it can help us make a lot of connections between things here. So if you were to think about it this way, right, this question, we have glucose, and we know that glucose can be converted to pyruvate. And in that process, and I'll just write out net ATP, so that would of course be glycolysis. In that process, there can be then a total of net ATP, 2 ATP made, right? Right now, in yeast, we didn't really talk about this, all right, but yeast will take pyruvate in an anaerobic environment and make ethanol from that. Okay, and there's a lot of big companies in Fort Collins that make a lot of money off of that whole reaction there, but the whole idea of this would be, of course, that in anaerobic conditions, right, this process will recycle that NAD+. Okay, and so that's what would be happening in the yeast cells. In the absence of oxygen, you get 2 ATP per glucose molecule, and of course you produce ethanol. Interestingly, this also produces carbon dioxide. We didn't really talk about this in class, but I'm going to go ahead and put this anyway. Carbon dioxide, so it'll make things fizzy and alcoholic. But anyway, okay, so so that's what's happening anaerobically. Now, when we look at this from this standpoint, of course, what we did in class is we talked about lactate fermentation. So mammalian systems, instead of making ethanol, they'll make lactate, but the same idea, okay? But when oxygen is introduced, all right, that will allow essentially this pyruvate to move, and it would require a transporter to do this. Okay, but it will move into the mitochondria, So let's just say that this is the matrix. Of course, yeast cells are eukaryotic, so they have a mitochondria. And that pyruvate then will be converted by PDC to acetyl-CoA. Okay, and then as we know, that acetyl-CoA will enter into the citric acid cycle. Okay, CO2 would be produced through this process. A small amount of ATP would be produced through this process, and a fairly significant amount of NADH would be made through this process. Okay, and then we have, of course, as we discussed in chapter 13, we've got, I'm just going to draw it as a single block, the electron transport chain, which will take this NADH, convert it back to NAD+. That will then drive proton movement, lots of protons across here into the intermembrane space, and then those protons will enter back into, this is ATP synthase. Okay, and it's got the little stator here. There we go. It's a high resolution structure of that. Those protons are going to go back through here, okay, and come through here. And that's going to allow that whole thing to make a whole lot of ATP. Right? So that's all the processes that occur when oxygen is present. Now, what I didn't do here, and I think that that's very important that I do this, oxygen is going to be here. So let me draw oxygen. Oxygen's converted to water. Right? And these electrons from the NAD plus or the NADH are going to make their way to that through the electron transport chain. Okay? And that's in aerobic conditions. Right? Now, the key here is going to, of course, be this ATP, this ATP. And it turns out that then in these conditions where there's oxygen present, this doesn't occur any longer. And for each glucose molecule, instead of getting 2 ATP, which is what you would get in anaerobic conditions, you get 30 to 32 ATP per glucose, okay? Depending on, again, how many C subunits are in the C ring of our ATP synthase, right? So the reason then that the yeast molecules will stop consuming so much glucose, or they'll reduce the amount of glucose that they are consuming, is because they're getting more ATP per glucose. In other words, aerobic conditions are a more efficient means of utilizing glucose. And because of that, then the yeast cells don't have to use nearly as much of the glucose molecules. Okay, so going back to our question here, let's see here, where is it? It's right here. Our question states, why would the yeast cells consume less glucose in the presence of oxygen? Because they get more ATP per glucose, okay, when oxygen is present. Right? Now, then it goes on to say this, why would DMP counteract the Pasteur effect? Meaning this, that you could put the yeast cells into the aerobic conditions, the more efficient way of using glucose and getting more ATP, but then if you add DMP into the mix, even if oxygen is present, the yeast cells will start consuming more glucose once again, okay? And the reason is this. Come back here. Where is it? There it is. The reason is this, because DMP, see if I can't get a good color here, DMP will take these protons, so I'll just draw DMP here. It'll take these protons and it will move those protons across the membrane. So we have DMP and I'll put one of the protons on it. There we go. It's not really what it would look like, but you get the idea. So DMP starts to take those protons that would normally go through ATP synthase, and it starts to move those through the membrane the inner membrane of the mitochondria independent of ATP synthase. And therefore, the protons don't produce as much ATP as they normally would, and glucose then doesn't produce as much ATP as it normally would. And so then the yeast cell, to maintain homeostasis in a steady state, has to consume more glucose once again, even though oxygen's present. Okay, so that's the Pasteur effect, and then that's what a DMP, like something like an uncoupler, can do. Okay, and of course DMP is one of those unnatural uncouplers. We talked about some natural uncouplers in brown adipose tissue in the Echo 360 recordings. So, okay, all right, so that's I think connecting, of course, some of the stuff that we've talked about with glycolysis, and this obviously connecting those concepts to the citric acid cycle, and then of course electron transport chain and ATP synthase, what we call oxidative phosphorylation. And it's this type of stuff that you're going to have to be able to do on the exam, right? So I've gotten several emails in the last couple of days saying, you know, what should we know about the citric acid cycle, or what should we know about electron transport chain, or what should we know, you know, these types of questions. And my response has been the same. It's been, well, you're going to want to know enough to solve problems like this, okay? Meaning that you need to be able to speak the language of glycolysis, you know, the citric acid cycle, and electron transport chain, and beta oxidation, those kind of things. And then you need to be able to make the connections that all those pathways have with one another, so that you can understand things like the Pasteur effect, which is what that short answer was all about. Okay. All right, so very similar to what I said with glycolysis in chapters 9 through 11. If you go back and look at the announcements in Canvas, I kind of made a post saying, okay, you know, straight memorization, not necessarily discouraged, but won't it be enough if you don't make the connections to be able to solve problems like the one we just looked at? Okay. Okay, so let's come here. I want to just make sure. Let's see here. Let's go to this. Just make sure the chat. Okay, we do have something. Does DMP poke holes into the membrane to allow movement of the protons? Good question. So no, it actually doesn't. DMP is a small molecule. I believe in chapter 13, I even give the structure of it. I can't remember what slide it is, but it's a small, actually fairly, it's hydrophobic, but it has these groups on it, these ethanol groups on it that make it fairly hydrophilic. But it's not, at least not to me. I don't know. I don't know if it's understood how it gets through the membrane. It's either being transported by a transporter, or it can go through there on its own through simple diffusion. In all likelihood, looking at the molecule, I would imagine it's being transported. What happens though, it's got these hydroxyl groups on it that can, depending on pH and low pH, it'll protonate, and then that can go across, and when it gets to high pH, like you would have in the inner membrane, then it will deprotonate. So, but it doesn't poke holes in it, no. It's going through one of the existing holes already. Okay, you're welcome. And then let's see, there's another question. Can you explain the math behind lipid catabolism of fatty acid, where the 6 electrons make 3 NADH and 7.5 ATP, compared to the glucose making 4 electrons would make 2 NADH and 5 ATP only? I'm not entirely sure what is being meant by this question in this. Maybe you could explain that a little bit better. Let me come back to that and try and explain that, because I'm not sure. It says where the 6 electrons make 3 NADH and 7.5 ATP, compared to glucose making 4 electrons would make 2 NADH and 5 ATP only. So glucose, you know, both glucose and fatty acid metabolism will make acetyl-CoA. That acetyl-CoA, once it gets in the citric acid cycle, will make the same amount of NADH. So it'll make 3 NADH per turn of the cycle, and 1 FADH2 and 1 ATP. So I'm not sure what you mean by that. Maybe either Madeline, maybe, you know, explain that a little bit more in the chat, or you can send me an email and we can talk about that too. So okay. Or if you want, you can just unmute yourself and ask the question that way. So let me come back here. Okay, so I've kind of cleared out that chat. Let me come back here now and look at the next question. What role do thioesters play in the PDH complex reaction, citrate synthase reaction, alpha-ketoglutarate, and succinyl-CoA synthetase? Very good question. So let's think about this. So when we look at thioesters, the two thioesters that we're talking about in this regard would be this, okay? So you have pyruvate that will be converted to acetyl-CoA. And I'm going to draw it this way. I'm going to put the S in there because this is a thioester. Acetyl-CoA is a thioester. Okay, and then you have another molecule later on that will be alpha-ketoglutarate that will become succinyl-CoA. And that is also a thioester. Okay, so these are our two thioesters that that last question was referring to. Okay, thioesters are great ways to store energy. And they store energy from the two reactions that are catalyzed to make these, right? So pyruvate to acetyl-CoA is catalyzed by the pyruvate dehydrogenase complex, and alpha-ketoglutarate to succinyl-CoA is catalyzed by the alpha-ketoglutarate dehydrogenase complex. Both of those complexes have really identical mechanisms. Their substrates are just slightly different. And so there's a lot of energy in these redox reactions that these complexes are facilitating or catalyzing. And that energy is captured in these thioesters, okay? So this thioester, let's deal with the bottom one first because it's probably the easiest and most straightforward. This is going to be converted to succinate. Okay, and it will make, I'll just put GTP here because that's what we have in most of our notes, but it could be ATP depending on what tissue type you're talking about in a mammalian system. But that thioester, its energy is going to be stored in this molecule GTP or an ATP equivalent. Okay, so essentially you have energy here that is transferred to a thioester that is then transferred into this GTP or ATP molecule. Okay, you also have here, and what's hidden here, and I didn't write this down, but what is hidden here is that NAD plus is converted to NADH. Okay, and this is an indication that we have a redox reaction going on, and that's where that energy comes from, is that redox reaction. Okay, now in this one, let's go to the top one because it's not really quite as obvious as what's going on in the bottom one. But in this top one, that acetyl-CoA molecule is going to combine with oxaloacetate. Okay, and then oxaloacetate molecule and acetyl-CoA molecule are going to become citrate. No ATP is produced. Okay, now here's kind of the trick with this one. The trick with this one is that this molecule here is in really, really low concentration in the matrix of the mitochondria. Okay, extremely low actually. And so because of that, and when you think about reaction and reaction rates, and also reaction thermodynamic favorability, when you have something that's in really low concentration, it will be thermodynamically not very favorable, right? Because you just don't have a lot of the reactant there. But this reaction, even though one of its reactants, oxaloacetate, okay, is in low concentration, it's still quite favorable. And the reason is because the amount of energy that's found in this thioester here, okay? And I think that I likened this in the Echo 360 recording that said that acetyl-CoA provides the energy to kind of get the cycle going. It's the kickstart. And I likened it to the idea of starting a lawnmower, right? You know, a push lawnmower is something that you grab the cord and you yank on the cord. And then when you yank it, you start, of course, the engine, usually, okay? So acetyl-CoA, that's the energy that thioester is going to be doing. It's just going to provide it to just kind of kick the start the whole thing because oxaloacetate concentrations are in really low concentration there. So, okay. All right. Let's come back here. I think that I may have gotten some. Okay, so come back here. Can you go through number 2 and 10 on chapter 13 study guide? I don't have the study guide pulled up. Let me see. I've got another computer sitting in front of me. Let me go ahead and pull up the study guide, and I'll do that as well. Just a moment. It won't take that long to get it. Okay. Okay, so this is in chapter 13, number 2 and 10 on the study guide. Let me so we don't spend too much time on one question. I'll come back to maybe number 10, but let me deal with number 2 first. How do electron affinities of each redox center compare with one another? In other words, would you expect redox centers in complex 4 to have a higher or lower redox reduction potential than the redox center found in complex 1? Why? What does that mean in terms of the delta G of the redox reactions? Okay, so that's actually a fairly straightforward answer. We expect that the redox or the reduction potentials of redox centers in complex 1 are significantly lower than that of 3 and 4. Okay, meaning that when you get to the very end of the electron transport chain, you have redox centers that are really high in reduction potential. Okay, in fact, oxygen, which is our terminal electron acceptor, has the highest reduction potential of all the redox centers found in the electron transport chain. NADH, which is the initial electron donor, has the lowest reduction potential of all of them. Okay, and there's a huge separation actually between them. I think that the reduction potential of oxygen is like 0.816. And of NADH, it's negative 0.320 volts. Okay, the importance of that is that redox reactions are spontaneous, meaning they're exergonic. They have negative delta G's when the donor has a smaller reduction potential than the acceptor. Okay, so take our extreme example here. Let's come back to this. Okay, if you think about the electron transport chain is just something like this. Let's just put it as a box, okay? And we have NADH as our redox, our electron donor, I'm sorry. It's going to give electrons here, and then this, as we know, those electrons move through a rather complicated process to oxygen, okay? And so we have all these things that go on here, all these reactions that go on that eventually make those electrons pass on to oxygen, okay? Well, this is our donor then, okay? And the reduction potential of it, like I said, is equal to negative 0.320 volts, okay? And then this is our acceptor. Okay, and this has a reduction potential that's equal to 0.816 volts. Okay, well, those two are really different from one another, but what that means is that we have a very negative delta G reaction, very thermodynamic reaction, getting electrons from NADH to oxygen, because the acceptor has a significantly higher affinity for electrons than the donor, okay, which has got a really weak affinity for them. All right, and if you were to trace that out at each one of the steps in complex 1, you would find out that as you move up, like let's say that chain of iron sulfur centers all the way to coenzyme Q, you would find that the reduction potentials of those go up incrementally until finally you get to coenzyme Q, and coenzyme Q has a larger reduction potential than NADH. So that's an exergonic then redox reaction in complex 1 when NADH passes its electrons to coenzyme Q. And that then exergonic reaction pumps protons. It causes conformational changes in complex 1 that allow protons to be moved from the N to P side. And that then stores energy in that redox reaction in a proton gradient that it then goes on to make HCP. So this is one big thermodynamic issue that we're looking at here, or a problem that we're looking at here, and that is we're taking essentially energy in one place and storing it in another place. Okay, so these redox reactions are exergonic throughout. Okay, so hopefully that helps you out. Okay, all right, so I'll come back to question 10. I'll just kind of keep going back and forth here between the chat and also the questions on the Q&A, but I'll come back and get to number 10 and some of these other ones that are being asked too. Okay. All right, so coming here now, We talked about the role of thioesters. Practice exam questions number 31 through 35. Let's do that. So I can come here. Come here. 31 through 35. Let's see here. Okay. Oh, okay. So we get the diagram more of this. So it says antimyosin A blocks electron transfer between cytochrome B and C1. If intact mitochondria were incubated with antimyosin A, excess succinate, and an adequate supply of oxygen, which of the following would be found in the oxidized state? Okay, so I'm gonna take a drink of water here just a second. Okay, so let's try and answer this by drawing this out between B and C1. Okay, great. So essentially what we have here, and this is, get to be a little bit more granular in our details here. So let's go ahead and draw out the electron transport chain as this. Okay, and so there's our 4 complexes. 1, 2, 3, and then 4. Okay. Okay, so let's do this here, and let's just do this. Okay, so we know this happens here. We get QH2 that gives electrons in this Q cycle, right? One of them comes back in this direction, but they're going to give electrons eventually. If I were to zoom in here, let me go ahead and erase this. I'm going to wait for that. This is going to give, in this direction, it's going to give to essentially, well, it's going to give to C1. It goes to this iron sulfur center, and then it goes to C1, okay? And then that goes to C. Okay, and then this goes to Bl, to BH, and then to another Q here. Okay, and so I'm diagramming some of the Q cycle there. Okay, but the question essentially says this. It says that this process is blocked. Okay, antimyosin A can block the transfer to C1 and also those B hemes. That's what it does. Okay, more than likely, if you're really wondering about this, is probably binding the QP site, antimyosin A. Okay, although that's a guess on my part. Okay, so then what's going to happen here is that kind of closes the door, if you will, in terms of the electron passage, meaning that electrons are not going to be able to pass any longer past complex 3, right? Now, presumably, and it says in the problem, I think that it says there's an adequate supply of oxygen, right? So there's oxygen that can allow for any electrons that had accumulated previous to antimyosin A inhibition. Any of those electrons can be moved through here to oxygen, making water, right? And then that leaves that door open, right? And then it also says this, it says succinate is bringing electrons to it, succinate, right? And those electrons are being passed eventually to coenzyme Q, okay? Now that process, right, will not allow any of this coenzyme Q is not going to be able to give electrons any longer to that, okay? And so what's going to happen is that this entire process is going to get backed up, right? So in other words, we can put little red X's all the way along here, okay? That would also be true of this. Okay, we should know this. NADH, of course, is giving its electrons, becoming NAD+, also to coenzyme Q. And so that's going to cause a backup, right? This is going to go back up this way, and this is going to inhibit all of those things here. All right, so then the question is, okay, well, what is the redox states of each of the things, each of the following, right, molecules? Okay, well, let's go ahead and look at this. All right, and what we see here, or it says, which of the following would be an oxidized state, meaning it can't lose its electrons, okay? and then it gives us a list of them. Okay. Well, coenzyme Q is the molecule that's going to be first affected by this. Coenzyme Q is receiving electrons from NADH ultimately and fructosuccinate ultimately, but it can't give electrons off anymore, and so therefore it's not going to be in the oxidized state. It's going to be in a reduced state because it keeps receiving electrons but has nowhere to put them because antimyosin A is preventing it from putting it somewhere. Right, so that would not be in an oxidized state, so we'd say false for that one, right? Not an oxidized state, it's in a reduced state. Cytochrome C is just on the other side of the Q cycle. Okay, cytochrome C is what's going to receive electrons from iron, sulfur, and C1, and then carry those electrons to complex 4. Well, that process is being blocked, so cytochrome C can't receive electrons, and therefore what will happen is that any cytochrome C that was reduced right when antimyosin A was introduced will go on and give its electrons to oxygen. That will clear it away, and all of cytochrome C will become oxidized. Okay, heme or cytochrome A3, that's in complex 4, right? So this is cytochrome C between complex 3 and 4. This is in complex 4. Same thing, right? So cytochrome A3 can't receive electrons any longer from complex 3. It'll give whatever electrons it had before antimyosin A was introduced to oxygen, and then it will obtain an oxidized state. So we'd say that's true. Okay, that would be true of complex of copper B, right? So these are both in complex 4. Okay, and then this is between 3 and 4. It carries electrons from 3 to 4. Okay, and then finally FMN. So copper B is going to obtain an oxidized state, just like cytochrome A3. But then this one, finally, FMN. FMN is the first molecule that receives electrons from NADH. Okay, so NADH, like we said, is everything's going to back up, and electrons from complex 1 we're going to keep trying to move them to coenzyme Q, but eventually coenzyme Q is going to say, well, there's not going to be enough of it to receive any more electrons because it's going to be all in the reduced state. And then everything in complex 1 and everything in complex 2 will obtain a reduced state as well. So FMN is part of complex 1, so it's not going to obtain an oxidized state, it will obtain a reduced state. Okay? All right, so hopefully that makes sense to those of you that were wondering about that question. Okay? All right, now I'm going to come back here. Okay, so it says, can you go through number 2 and number 10 on chapter 13 study guide? And we also got this one hanging out here. Go over number 13 on chapter 13 study guide. So let's look at these questions. What would happen if no oxygen were present? Okay, how would this affect the citric acid cycle in the PDH complex? What would happen to NADH levels in the mitochondria? Would they go up or down? How would this no oxygen affect the amount of glucose catabolized by glycolysis? Okay, so this goes right along with that set of questions we were just looking at. Okay, so let's come here and let me come back to our diagram here. Okay, so what this is essentially saying, let me get rid of some of this. Go here. Get rid of this. Get rid of this. Get rid of that. Oh, I just got rid of all of complex 2. That's not what I wanted to do. It's all right, I can put it back. Okay, good deal. There we go. Okay. Oops. Okay, so let's get rid of that. Okay, I'm just going to get... Okay. All right, so we've got our electron transport chain. Okay. And essentially, the question that's being asked this is number 10 on chapter 13 study guide. What would happen to this whole thing if oxygen is not present? Okay. And so essentially, we're just kicking the can up a little bit. We just talked about antimyosin A. Well, with oxygen, we're just removing this, right? And then what that does is, if you think about these electrons, they're passing here, they go through here, they go through here, right? And what that does is essentially, it just inhibits all these processes, right? And this whole thing is going to obtain a reduced state. all the steps in it, okay? And we don't want to go over the nauseating details of all the steps, right? But I mean, you're right, in complex 1, you have what, 7 iron sulfur centers, or 7 to 8 that accept electrons, that kind of stuff. Right, all of those are going to obtain the reduced state. Okay, and you got the Q cycle, and you got all the stuff in complex 4. Okay, that's going to do this. And you can also think about it this way, right? Succinate gives its electrons, and then it kind of comes into this whole process. Same thing. It's going to back this whole thing up, back. Okay, the whole thing jams up. Okay, now the question goes on to say this is what would happen if no oxygen were present? How would this affect the citric acid cycle in the PDH complex? Okay, well, let's come here and let's look at that. Okay, and so we've got the citric acid cycle. Let me just go ahead and draw this. We got pyruvate converted to acetyl-CoA. Okay, and the acetyl-CoA is going to enter into that cycle, and then the cycle is going to turn like that. Okay, and we got several points along the way. Okay, that make NADH, that make FADH2, that make ATP, that kind of thing. Okay, well, it turns out that NADH is an inhibitor, okay, of the PDC, pyruvate dehydrogenase complex. It also inhibits citrate synthase, so it will stop this process, and it also inhibits alpha-ketoglutarate dehydrogenase. Okay, complex. So it inhibits 3 of these points that we're talking about here, meaning that all of a sudden flux through the citric acid cycle, when this electron transport chain stops running because there's not oxygen, flux through the citric acid cycle in the PDC is going to really slow down. It's going to kind of grind to a slow state, okay, or very slow flux. The question then goes on to say this: what would happen in NADH levels? So we talked about this. NADH levels, by the way, I didn't really say this. I should have said this. This is where I should have started. NADH levels are going to go up, right? Because they can't be recycled. NADH is being made by the citric acid cycle, but they can't be recycled by the electron transport chain, okay? Because the electron transport chain is inhibited. So the citric acid cycle will keep making that NADH, but that NADH has nowhere to go to. So they'll feed back and inhibit the citric acid cycle. Okay, so NADH levels go up, citric acid cycle flux goes down. Okay, and then how would this affect the catabolism of glucose by glycolysis? Well, what would happen out here, right? This is all happening, everything that we talked about in here is happening in the matrix of mitochondria. Well, eventually pyruvate, right, would start to back up because pyruvate's not being used by this process, and that would feed back, okay? And that will actually start to be converted to lactate, because the cell is going to start to lose out ATP, because you're losing all of these processes that will normally make ATP, and ATP is going to ATP would normally kind of slow glycolysis down. Okay, well, glycolysis is not receiving that feedback. Like, there's a lot of ATP being produced by aerobic respiration, because ATP levels will start to drop because you removed oxygen, and that will increase flux through glycolysis, and the pyruvate will start to be converted to lactate really rapidly, and glycolysis will start to go through actually a really large increase in flux, okay, because the low amount of ATPs that's being made will activate that pathway, okay? All right, so hopefully that will make sense, and see how all that's connected. And this is actually directly connected to the question that we talked about at the very beginning of our review session, and that was the Pasteur effect. Okay, essentially, what's happening here is, if we were to introduce oxygen, right, then you could open up these pathways to produce more ATP. Okay, per glucose molecule. Okay, all right, good deal. Now let me real quick. Let's just see here. Okay, let's go to our next question on this. Okay, how could OOA levels be restored to steady state levels? One reason OOA levels in the mitochondria could decrease. It's a great question. Let's go ahead and look at this. Okay, the citric acid cycle. Okay, I won't draw the whole thing in gory detail, but we have acetyl-CoA, and it's going to combine with oxaloacetate. Okay, so it combines with oxaloacetate and it becomes citrate. Okay, and then that's through a couple of steps. Like I said, I want to draw the whole thing in real gory detail, but that's going to make some alpha-ketoglutarate. glutarate. Okay, and that will make succinyl-CoA. Okay, and then we'll make some succinate. I'm going to go ahead and draw a couple of these here because they're important for this little process here. And that will make, through a couple of steps here, malate. That can be then converted back to oxaloacetate. Okay, all right, so that's a decent enough way of doing this. Now the question is, let's come back to this. If you look at it, how could levels of OAA be restored to steady state levels? One reason OAA levels in mitochondria could decrease. So let's do the decrease first, and then we'll talk about restoration. All right. And there's lots of reasons that OOA could decrease, okay? But if you imagine acetyl-CoA is feeding into this process, and every time acetyl-CoA feeds into this, a small amount of oxaloacetate is used, okay? And as long as those carbons make it all the way back around, nothing happens to oxaloacetate, right, in terms of concentration. For every one that's used, another one's produced. However, that's not always the case. Okay, so for instance, you could have one acetyl-CoA come in here, combine with one oxaloacetate, and become citrate. But that citrate could be removed from the mitochondria for other purposes. Okay, for instance, fatty acid synthesis. Okay, an anabolic process. Free fatty acids, not FAA, but F, free fatty acids, FA. Okay. All right, so that's could be something that happens. Well, that means that oxaloacetate that was used to do that, to make the citrate, is not going to be regenerated. So we've dropped oxaloacetate, right? Another one could come in here, right? Acetyl-CoA can combine with oxaloacetate, makes it to citrate. Oh, but it goes all the way past it. That's fine. But then all of a sudden, this has to be used to make glutamate for some reason. And that could be used to make glutamine, okay, or something along those lines. And you get the idea that there are things that can be taken away from the cycle. And when they're taken away from the cycle, that will cause a depression in oxaloacetate. Something else that we've talked about, of course, in this class, going back to chapter 11, is that oxaloacetate can be used to make glucose, okay? So there's lots of things that will start to take that oxaloacetate away from the cycle. And when that happens, what can happen is that the entire flux of the citric acid cycle will start to drop, because really what's kickstarting the whole thing, right, remember, is oxaloacetate combination with acetyl-CoA, right? So in those cases, and understand that I'm drawing several options here. All of them are more than likely not necessarily happening at the same time. They could be, okay? But those are going to start to depress flux through the cycle. So that's what could cause oxaloacetate concentrations to go down. So then the question is, well, how would you get that back? Okay, and there are actually several ways that can happen. Okay, let's look at two of them here. Okay, and I will use let's use green. Green's a good color. It means like that seems like you're adding something back. So one of the ways that this can happen is this: pyruvate, as we know, can be absorbed by the mitochondria, okay? And pyruvate can be converted to oxaloacetate simply by the addition of bicarbonate, okay? It takes some ATP to do this, but the enzyme, if you remember, going back to your gluconeogenesis notes, is pyruvate carboxylase that will do this, okay? So interestingly, pyruvate carboxylase is an enzyme that certainly kickstarts gluconeogenesis, but you will find pyruvate carboxylase in lots and lots of tissue types that don't carry out gluconeogenesis. Okay, for instance, skeletal muscle, or cardiac muscle, or brain tissue, or we can just go through lots of these tissues that don't carry out gluconeogenesis. They don't have all the enzymes to do it, but many of them will express pyruvate carboxylase. And the reason that they have it is so that they can restore oxaloacetate concentrations to keep the citric acid cycle running, okay, for lots of very important reasons. Okay, so that's one way to do this. Another way to do this is to do the exact opposite of what's stated down here. And this is one of the more common ways of doing this. The two that I'm really going over, pyruvate carboxylation and the breakdown of glutamine and glutamate to alpha-ketoglutarate is another one. Okay, so take these two amino acids, and they're really abundant actually in our tissues, and break those two down into alpha-ketoglutarate, and that will then produce oxaloacetate, okay, through this pathway. It's reintroducing something to the pathway. Now, there are other ways to do this, but those are two of the very common ways to get oxaloacetate concentration back up to level, right? So let's say some cell, for whatever reasons, carrying out fairly significant amount of fatty acid production, okay? That cell can replenish oxaloacetate concentrations by simply carboxylating pyruvate, which many of them do, or by breaking down glutamate and glutamine. Okay. Now, interesting. Let's try and make this connection for you, right? Let's say that you're in an adipose tissue. Okay. Of course, right? Fat cells, and you have a high glucose content diet, right? Well, that glucose can be absorbed by your fat cells, right? And that glucose can be converted to pyruvate, and then that pyruvate can be converted to acetyl-CoA, and then that acetyl-CoA can be converted to citrate, and citrate can be made into free fatty acids, right? Okay. So in those cells, they'll have to have a source of oxaloacetate. Some of that pyruvate, instead of being converted to acetyl-CoA, will be directly converted to oxaloacetate. Either way, they're going to be, lots of it's going to be used to make free fatty acids, and of course that will be stored as triacylglycerides in those fat cells. So anyway, okay. Let's keep going here. Oh, this is so interesting. Awesome stuff. My wife is in the other room. She just laughed at me. So, okay. She thinks I'm a geek. I don't know why, but anyway. All right, so now she's really giggling. Okay, but anyway. All right, so can you please go over number 13 on the chapter 13 study guide? Let's do that. How much energy does it take to move a proton in the conditions found in the mitochondria? How much energy is available in the movement of electrons from NADH to oxygen / succinate or FADH2 to oxygen? How does this compare to the energy required to move the quantity of protons seen in each of these complete transfers? Okay, yeah, let's do that. Let's see here. Okay, so once again, I'm going to go ahead and kind of represent the electron transport chain as one big block, okay, that would encompass all of the complexes, okay, and we have here, let's actually, maybe I should have drawn it in two. I'm gonna go ahead and this will be all right. I think that if I draw this will be okay. All right, so what we have here is this, okay. I'm going to go ahead and put complex 1 right here, and then we'll put complex 2 right here, okay, and then complex 3 and 4, we'll put it here. So I can't draw it quite as a big block for this, right? But what we know here is this, okay, that complex 1 pumps 4 protons, and then complex 3 and 4 together are going to pump 6 protons, okay? This would be 4 through complex 3 and 2 through complex 4, okay? Complex 2 doesn't pump protons at all right? And when we look at this, all right, the delta G of moving one proton, okay, is equal to, so this is, actually let's do this. Energy per proton. I'm going to do this as being N to P. Okay, because this is the N side, this is the P side. So energy per proton pumped into P side is going to be about positive 21 kilojoules per mole. Okay, I wouldn't expect you to be able to calculate that although it's not too terrible to do that. I think the book tells you how to do that, but in this class, we'll just say that it's 21 kilojoules per mole under most conditions on average. Okay, all right, so that's how much it costs to move a proton across. All right, now what that means is this: when we think about this, right, these 4 protons here plus these 6 protons here, this is going to equal 10 protons moved when we add these 2 together, which means that this is going to cost about 210 kilojoules of energy, right? Because this is 21 kilojoules per proton pump. There's 10 protons being pumped, so that's 210 kilojoules, okay? Of course, if we were to look at only these 6, let's look at this, only these 6 protons, that's going to be about 150 kilojoules per mole. Okay. All right, so that's how much energy. Let me just real quick here. I got to check on my math just to make sure. Nope, not 150. I knew I looked at that. I said that's wrong. It's 120, 26. Sorry about that. I'll give myself a red check minus for that. Bad. Okay, there we go. So 126 kilojoules per energy, or per 6 protons pumped. Okay, so that's how much it takes to move that amount of protons. Now, here's why I did it this way. Okay, when we think about this, I use the same colors here. NADH brings electrons into here, as we have been saying, becomes NAD plus. And those electrons go all the way through here, skip over complex 2, go all the way through here, and then finally make it to oxygen. Okay. And that makes water. Okay. Well, when we look at the reduction potentials of NADH versus oxygen, which I actually talked about in this review already, right? It's negative 0.32 volts for NADH and it's 0.816 volts for oxygen. We do the arithmetic, and again, I'm going to ask you to do that on this exam. Okay, but when we do the arithmetic, that redox reaction, moving electrons from here to here, this is going to give, actually, let me really draw it from this arrow. It's really that whole process there gives 220 kilojoules of energy, meaning that its delta G is like negative 220 kilojoules per mole. Okay, well, that's enough to move 10 protons, and that's exactly what NADH does, moving electrons through complex 1, 3, and 4. It moves 10 protons. There's enough energy there to then move those 10 protons. This then efficiently stores, right, 210 kilojoules of energy of the 220 available. Okay, now succinate, on the other hand, succinate has a higher reduction potential than NADH. Okay, and it becomes, hopefully you would remember, this becomes fumarate. Okay, and that pumps its protons through here and then it connects in there. Well, that only moves 6 protons. Okay, well, if we were to look at that process based off of its reduction potential of succinate and oxygen, this gives, this is where I got my 150. This gives about 150 kilojoules of energy. Oops. Okay. In RG, not NGR, energy. Okay. And that moves, that's obviously enough to move 6 protons. Okay. Actually, a little bit more than 6 protons. It does move 6 protons, but it's storing this energy, right? Or I'm sorry, these protons, 6 protons, are storing 126 of the 150 kilojoules of energy available in that redox reaction. Okay, so that's what that question on that study guide is talking about. How much energy does it take to move a proton? It's 21 kilojoules of energy. How much energy is available in the movement of the electrons? Right, 220 kilojoules of energy for the movement of electrons from NADH to oxygen, and 150 kilojoules of energy available in the movement of electrons from succinate to oxygen. How does this compare to the energy to move the quantity of protons seen in each of these complete transfers? Well, that's 10 protons at 6 protons. Okay, so that's essentially what we're looking at here. Okay. Okay. again, that's so cool, right? I mean, that's so cool that it's just all this energy storage. And then you think about this, the protons, and we talked about this in one of the coolest molecules ever, is ATP synthase. It takes that energy in the proton gradient, and that energy then drives conformational changes in this protein that are amazing, and that produces this ATP, right? And so all this energy is being used to make that ATP, which then can go on and allow us to contract our muscles and all that cool stuff. Okay, all right, I found it so cool, I know. People laugh at me for that, but anyway, I'm a geek, what can I say? All right, now, what step of ATP synthesis requires energy when F1 is present? Great question. So that gets right to this idea of what I was just talking about, how the protons are going to cause conformational changes in ATP synthase that's going to drive the rotation of the C ring that's going to eventually lead to the production of ATP. When F1 is present, the step that needs energy to make ATP is actually removal of ATP from the active site. Okay, so it's not the production of the phosphate anhydride bond between ADP and inorganic phosphate, but rather it's the energy needed to kick the ATP molecule out of the active site, and that process is done by these conformational changes, right? As that C ring is rotating, it's rotating that central stalk or that gamma shaft, and that gamma shaft is literally running into the ATP molecules in the active site and forcing its way out, or forcing the ATP out of the active site. Okay. Let's see here. Let's come back here. All right. So I don't have any more questions there. It's 1:56. Let's take another 4 minutes. We'll go straight to 2 o'clock, or I guess it's 1:58. So take another couple minutes, try and answer some of these other questions, and then I'll let you all go. Okay. So does complex 3 pump 4 protons to the p side as well as 2 protons to the inside? No, it doesn't. So it pumps 4 protons total to the p side. It takes up an additional 2 protons from the inside as it does this, but it only pumps 4 of them per cycle, right? Each round pumps 2, okay? And then each round picks up 1 additional one from the inside, okay? But we only count it as 4 total pumps, okay? All right, number 13 through 15 of the practice exam. Let's see. I'm gonna look at these other ones. Okay, so number 13 through 15. Let's make this our last one, okay? And then I'll let you all go about your ways, because you have other exams to study for too, I'm sure. Okay, entry of acetyl-CoA in the citric acid cycle can decrease when NADH is rapidly oxidized through the respiratory chain. Okay, so the key word here is decreased, right? Entry of acetyl-CoA into the citric acid cycle. So when NADH is rapidly oxidized through the respiratory chain, let's come back to some of our diagrams. Let's see, we have one right here, I believe. This one is a good one, I think. Okay, so well, obviously this is an inhibition of all of this, but if you were to consider when none of these red X's are here and there's lots of oxygen and there's a great need for ATP production, this NADH can be used very rapidly in this process. All right, and when it's being used very rapidly, it's sucking that NADH away from the citric acid cycle, and that will increase the amount of acetyl-CoA going into the citric acid cycle. Flux will actually increase. Okay, so NAD plus, which is, of course, the product of this can come back, and this can feed back and activate this process. So if there's a lot of NAD plus production, then that can activate the citric acid cycle, which means it will start to draw in acetyl-CoA very rapidly. Okay. Okay. Next question, 13 through 15. The energy charge is low. Okay, so when energy charge is low, what that we're saying is that ATP levels in the cell are low. And when ATP levels in the cell are low, then that's going to activate catabolic pathways. It's going to activate glycolysis, and going to, as long as there's oxygen present, it's going to activate the citric acid cycle. And so then if the citric acid cycle is activated, that means that acetyl-CoA will be used more rapidly. The opposite is true in 15. When the energy charge is high, that means there's lots of ATP. That means that ATP is going to start to inhibit the citric acid cycle. And then that's going to decrease the amount of acetyl-CoA going into it, because citric acid cycle flux will drop. Okay? All right. So really, just out of respect to your time and these kind of things, I'm going to go ahead and end our time together. Okay. I have really enjoyed this semester. I really have. It's been a really unique semester, but I've enjoyed getting to know all of you through email and through office hours and through all the other avenues that I've been able to get to know you. So we have until tomorrow. So if you had questions that were unanswered, you're like, man, I really needed an answer to that, go ahead and send me an email, okay? And I'll try and get those questions answered before you take your exam tomorrow. Good luck in obviously BC351 exam, and also good luck in your other exams for your other finals. Okay, so and then you have a great holiday season, right? A Merry Christmas and a Happy New Year and a happy holiday season. Okay, all right. I think that's it. So take care, everyone. I'm going to go ahead and stop the recording now. Right. One question: will you add the chapter 14 annotated slides? And yes, I will. I'll do that. So okay, all right. Take care, everyone.